So I must congratulate and appreciate the organizers to include the food safety in the theme. Most of the time we talk about food security, but we often ignore food safety. So I must congratulate the organizers to the inclusion of food safety as one of the important focus area in the theme. So here what we are doing at ICRISAT, I'll give you a snapshot, especially on this particular problem. And so just before going into my presentation, just a minute about our institute, that is ICRISAT. Those who are not aware of ICRISAT, ICRISAT is an international crops research institute for the semi-arid tropics. Established in the year 1972, there were two foundations played a key role, Rockefeller Foundation and Ford Foundation, in the establishment of ICRISAT in India, with a huge support from the government of India in 1972. And we are a non-profit, non-political organization, agricultural research for development organization, and we have a mandate to work in the semi-arid tropics of Asia as well as sub-Saharan Africa. And we work on 6 mandate crops, especially 3 cereals and 3 legumes. The cereals include sorghum, pearl millet and finger millet, and the legumes include chickpea, pigeon pea and peanut. And we have our global headquarters in India, in Hyderabad, in Telangana. And then we have very strong presence in several countries in Africa, both in the East and Southern Africa region as well as West and Central Africa region. So this is briefly about ICRISAT. And before going into my presentation, I just want to give you another update. I don't know how many of you heard about this incident, which happened exactly a year ago in India, last October 29, 30, 31st. In the central part of India, in Madhya Pradesh, there were 10 wild elephants entered into one kodo millet crop area, and they fed on the crop. The crop is heavily infested with a fungi, and a lot of mycotoxins were there on the grain, and within 3 days, 48 hours, 72 hours, 10 elephants were died. So this has become a big national issue, and had a discussion in the Parliament of Government of India, and they constituted an investigation team, and I'm one of the part of the investigation member, and several samples after the post-mortem received to my lab, and we found that huge quantities of cyclopyrazonic acid found in the gut of the dead animals. Cyclopyrazonic acid is also one of the important mycotoxin, often ignored, and produced by the fungi like Aspergillus, Penicillium cyclopeum, and other Talaromyces and other fungi. So this particular incidence underscores the importance of addressing mycotoxin contamination in food commodities. So I'm not going to discuss about this particular issue because we just started working on this issue, and our first investigation paper published last week only. This got approved in the Journal of Wildlife Science. So coming to the actual topic, aflatoxins in agriculture commodities. And this is one of the world's most important qualitative issue and affecting many crops. A few important ones are like groundnut, maize, and several tree nuts. And first came into limelight in the year 1960, especially in the United Kingdom, when hundreds of thousands poultry birds were died after fed with contaminated meal which imported from Brazil, peanut meal. And then the work started. Know what was the problem and what was the death of the poultry birds, and by the 1966 the researchers able to address that. The reason is the aflatoxins produced by a fungi called Aspergillus flavus. That's why the name has come, aflatoxin, Aspergillus flavus produce toxins. Of course later there were many toxins were identified, but these are the few important ones: B1, B2, aflatoxin G1 and G2. B stands for blue fluorescence and G stands for green fluorescence because they have the kind of fluorescence property when you observe under the fluorescence light. And the question is, and aflatoxin M1 is also an important, especially the animals when fed with contaminated feed, the milk is also getting contaminated by aflatoxin M1. So the question is about what is this issue? Is it an economic issue or a health issue? So when, especially in developed countries like in the United States, they spend millions of dollars to ensure that the public safety as well as to ensure that the value chain is clean and no contaminated food and feed enters into the chain. So it's an economic issue for them, but that was not the case in especially in developing countries as well as less developed countries in Asia and Africa. So it has become a health issue in all these countries. So if you look at the effects of these, not just the health effects, it is also affecting hugely the trade. Actually, the trade prospects are also getting affected by the contamination of aflatoxins in food commodities in groundnut. And very recently, there was an evidence that all these years, liver cancer, hepatocellular carcinoma, one of the predisposing factors is aflatoxin contaminated diet exposure. But gallbladder cancer is also linked to the aflatoxin contaminated diet. That paper has been published very recently from India 2025. So you can access that paper and read. I'm also part of that investigation. So if you look at the regulations worldwide, keeping in view of its negative health as well as trade effects, several countries, more than 100 countries in the world are imposing stringent regulations on these aflatoxins. European Union is one of the most stringent, the list and the countries list, imposing very severe regulations. Any commodity which is more than 4 microgram per kg, they won't allow for direct consumption. And there are other countries which are having 10, 15, up to 20 microgram per kg. So just this is one sharp snapshot of India's peanut exports. If you look at India's peanut export scenario, India is unable to export peanuts to the European Union just because of this particular reason, very stringent regulations of aflatoxins. We could be able to send our peanuts to other countries where slightly lower regulations were in place, like Indonesia, Vietnam, Philippines, but our exports to European Union is almost nil. So even though European Union is one of the big niche market for quality products, but we are unable to export because of this particular issue. So if you look at what are the most important predisposing factors for this problem, especially in groundnut, I would like to focus on groundnut. So the first and foremost reason is the end of season drought, and then the soil health issues, and then the insect damage as well as the nematodes and termite damage, and the improper drying and storage after harvest, and also the varieties, which are all important predisposing factors for this particular problem. And the interventions can be divided into two, primary and secondary, but we don't talk about secondary interventions, only we focus on the primary interventions, which are agriculture interventions. So we at ICRISAT, we work in several dimensions to address this particular problem: host plant resistance, biocontrol aspects, and also some of the agronomy practices with which we can able to reduce this particular contamination. But overall, not only at ICRISAT, globally, the limited success has been noted especially in case of host plant resistance. Even today, we don't have a kind of varieties which are highly resistant to pre-harvest aflatoxin contamination. So with that, actually, we started working on one unexplored area, that is seed coat, because there are two important physical barriers to enter the fungus into the groundnut kernels. One is the shell, and the second one is the seed coat. So the fungus has to cross these two important physical barriers to enter into the kernel and then produce the toxins near the cotyledon. So we want to focus on the seed coat, and we want to see what kind of biochemical composition is helps to stop the fungus growth as well as toxin accumulation. So here we have selected two important varieties. One is known as resistant after several field trials, that is 55-437, and one is highly susceptible, that is TMV2, and we want to deeply understand what kind of biochemical composition differs between these two varieties. So when we first started this experiment almost 6-7 years ago with the help of USAID Peanut Innovation Lab project, so in the TMV2, when we remove the seed coat, so when there is no seed coat, you can see the full of fungal growth. When there is a seed coat, even though it is a susceptible, the fungal growth is limited actually. So the same in the case of 55-437, which is known resistant variety. When you remove the seed coat, you will see the fungus growth fully, and when there is a seed coat, intact seed coat, you will see very less fungal growth on the seed coat after challenging with the Aspergillus flavus toxigenic strain as well as non-toxigenic strain. This clearly giving us a kind of glimpse that yes, there is something there on the seed coat, and which is stopping the fungal growth as well as its proliferation on the seed. And also you can see many-fold changes when compared to the susceptible variety in terms of the fungal DNA present on the seed. So the second experiment is we thought of looking at the soluble and insoluble peanut seed coat biochemicals from these two varieties after challenging with the toxigenic one. And again, we found very interesting results that there is a significant differences between the resistant and susceptible one in terms of extracted soluble and insoluble seed coats, which are inhibiting the fungus growth. And important thing is seed phenolic compounds. We started looking at the seed phenolic compounds present in these two varieties, and there were several phenolic compounds which were significantly different, but one which attracted to our attention is the ferulic acid, which is significantly high in the resistant one compared to the susceptible one. And there was some literature showing that ferulic acid is an important fungal growth inhibitor. And then we started looking at the ferulic acid. When you go on increasing the concentration of ferulic acid twofold, threefold, fourfold, we have seen a tremendous inhibition in the growth of the fungus. So that means this particular phenolic acid is playing an important role in the seed coat in stopping the fungal infection as well as toxin accumulation. And also we have looked at the phenyl propanoid related genes, which have shown again significant expression pattern differences between these two. And one of the most important thing is the lignin monomers actually. So there was no significant difference among the total lignin content between susceptible and resistant one, but there is, if you look at the lignin monomers, H-lignin is one important one. There is a significant difference between these two varieties. That means again lignin is also playing an important role in stopping the fungus growth as well as toxin accumulation. So overall, we found that the intact seed coat protects and inhibits the A-flavus colonization, especially when we understood both the resistant and susceptible one. And also there are several other evidences which are supporting our hypothesis that seed coat is very, very important in stopping the fungus, so that we can look at the seed coat, how we can strengthen, how we can manipulate the biochemical constituents so that we can reduce the infection. And further, we even identified some of the important genes, candidate genes, which are linked to this particular resistant one. And also the secondary cell wall thickness is very, very important. As I mentioned, the lignin deposition is playing very important role in the case of resistant one compared to the susceptible one. And clearly you can see the histochemical analysis. The more and more deposition of lignin corresponds to the less and less inhibition by the infection by the fungus. So, and then we need to look at we need to look at the developmental biology of the seed coat. That is very, very important, because the seed coat is not going to be formed in 1 day or 2 days. Actually, there is a huge process, takes weeks of time for the formation of seed coat. We need to better understand at what time this lignin deposition is happening, so that we can able to look at the manipulation of this particular seed coat. And then we also work on several other aspects, not only host plant resistance, like water management is very, very important one, like how best we can able to keep the soil field moist for a longer period, especially before harvest. And then you can see the mulching plays an important role, which keeps the soil moist for longer duration and thereby reduces the pre-harvest aflatoxin contamination. And then we also work on storage aspects, especially drying and storage. And then the drying methods always important. The storage is one I want to highlight is hermetic storage, which is really gave us very good results in stopping the mold fungi infection as well as insect damage during the post-harvest, because of hypoxia and hyperparvia conditions when you store your groundnuts under in these kind of hermetic storage bags. This is one. Purdue improved crop storage bags, PICS bags. We have been promoting these bags after thorough evaluation. We found that they are very, very effective in keeping the quality of groundnuts. And this has been published in paper. And the diagnostics is another important one, because most of the diagnostic tools that we are currently using in the laboratory are cumbersome actually, because you need to bring the sample to the laboratory and you need to crush the sample, you need to extract the toxins to quantify either using chromatography methods or immunoassays. But there is a need for the non-destructive and rapid methods of aflatoxin destruction, which is need of the hour, and the research has to be now focused in that direction, especially for the thing. And few other important areas are, like yesterday Dr. Andy Kumar mentioned about microbiomes. That is also one unexplored area, especially to address aflatoxin contamination. We need, rather than focusing on one or two important biocontrol agents, we need to holistically understand the microbiome vulnerable community structure and its role on the pre-harvest aflatoxin contamination. So that's also one unexplored area, and we need to look at it. And another thing is about biochar and other some soil cake soil amendments is also we need to better understand how it can reduces the populations of these toxigenics. So that's all, and I thank our especially donors who supported our work, USAID. But unfortunately, USAID innovation labs has been closed starting this year. We were unable to take this work to the second phase. Yeah, thank you.
Long live India and France friendship.